I'm delighted to welcome Professor Warren Tate. It's wonderful to have you here with us today. Many of you will be familiar with Professor Warren Tate and the enormous contribution he has made to medical research of ME and the validation of our illness. So he's going to speak with us today about DNA research of MECFS patients, and he's going to touch on the hottest topic of the community at the moment in terms of the COVID vaccine for us. So Professor Tate is an emeritus professor, and he's had a research career spanning 54 years at the University of Otago in Dunedin. He's a fellow of the Royal Society of New Zealand, a fellow of the Alexandra von Humboldt Foundation, and was an international research scholar of the Howard Hughes Medical Institute of the United States. He was awarded the University of Otago Distinguished Research Medal in 2006, the Rutherford Medal in 2010, and the Marsden Medal in 2018. He initially became a discovery molecular biologist with a fundamental interest in how proteins are made and the origin of protein synthesis and genetic code. He co-discovered a new mechanism of gene regulation used by the HIV-1 translational frame shifting. Currently, he's using DNA, RNA, and protein omics to understand the molecular basis of unexplained diseases like Alzheimer's. He's got a particular interest in the biological basis of ME and now long COVID, not only as a researcher, but also as part of that MECFS family. So we are extremely grateful and proud to have you as part of our community, Professor Tate. So I will stop my screen sharing and I will hand over to you.
Thanks very much. I'll now share my screen, hopefully. Okay. Take that off. Can you all see my screen now? Good.
Yes.
Yes, thanks.
Good, thank you. So thanks so much, Kia ora, or thanks so much for inviting me to join you today. I come here as a biomedical researcher, but particularly as an ME family, member of an ME family. And I must say I'm very humble just listening to all of the things you're doing now for ME people and you know, the community and you know, that's what I'm trying to do from my perspective as well. So what I'd like to do today is to talk to you firstly about what I see the last 18 months has done for MECFS and some of the things that might happen in the future. And then I'd like to talk to you about my own research, which had an initial aim of strongly affirming the biological basis of MECFS, then trying to kind of unravel the dysfunctional physiology that's occurring, that's to give the myriad of symptoms that you know you all suffer from, and then hopefully open some avenues where we can ameliorate the effects of this disease, you know, in the future. And so that's where I come from. So if I can go to I'll go to full screen. Okay. So how's the pandemic impacted on MECFS? And I want to reinforce the comments that Kate made. Actually, the SARS pandemic has made clinicians worldwide and the general public realize that actually with a serious infectious disease, then could come ongoing disease of post-viral fatigue syndrome. And of course, came rather late in the day, but they realized there's one already exists in the community, namely MECFS, which is a classic example. For me, in 2019, before the pandemic, I was really excited because the National Institute of Health in the United States, the institution that does the most research in biomedical research and gives more money for research funding, which had virtually given no money for MECFS research, promised that they would actually put $2 or $3 million into MECFS. I thought that was absolutely marvelous. But of course, this year with long COVID, we've now had a promise of $1,150 million being put aside to investigate the post-COVID-19 conditions, as it's now called, or long COVID. And of course, I'm very optimistic that MECFS research will be alongside that. And the good thing in the US is that there are organizations moving to keep MECFS and long COVID sort of linked together, and one like SolveME, for example. So I hope that actually a lot of researchers will now flock to this area. There'll be much more activity in trying to understand the disease. And, I hope in the relatively near future, we can see some amelioration of the condition. I'm also aware of the huge dilemma that MECFS patients face now about should I get vaccinated? I get many inquiries on a weekly basis now from people telling me their histories of ME and asking me whether they should get vaccinated. And while I would never tell someone they should or shouldn't get vaccinated, I, of course, try and tease out the risk factors for them. But I should say that some of them say they're under enormous pressure from their families and their friends and neighbours to get vaccinated when they themselves feel it's not in their best interest. And certainly with ME, with a disturbed immune system, with these sensitivities to chemical sensitivities, allergic responses, it's quite okay for ME patients to make the decision not to get vaccinated on medical grounds. I think that's a very reasonable thing to do. I should say just yesterday, something came into my mailbox, and it was the results of a study in Sweden involving 1.7 million people, where they were able to show that in a family where there had to be unvaccinated people and children who were not eligible for vaccination, having one vaccinated person in the family protects the unvaccinated by 65%. And if it's a big family and there are four vaccinated people, then the unvaccinated person is protected by 90%. So it's as good as having a vaccine. So that's quite a promising thing. And that's why I'm very supportive of vaccination for people, for family members of MEC patients, so they can be protected. And we hope that that will continue to carry on. So I wanted to say just a little bit about the Pfizer vaccine, because there are a number of sort of things going on around about it. Firstly, that this is a new technology, RNA technology, and so is this really a safe vaccine for us to be having, and wasn't it rushed through? Now, I'm actually an RNA biologist of old, so I was very excited to hear when the RNA technology has been worked on for vaccines for well over 10 years. And so the fact that it was ready to be used for the development of a vaccine was quite exciting. And one of the excitements is it's very easy to modify the vaccine if a virus changes its own structure. And so as is happening now with SARS, new vaccines can be established with the RNA vaccine technology, which can rapidly recognize new variants. So that's a plus. The diagram there is actually what the vaccine kind of looks like. It's in the middle, you can see a small piece of RNA, and that's a code for the viral spike protein, just that small spike protein. It's surrounded by a lipid nanoparticle, and that's there to allow this to fuse to our cells when it's injected, and so the RNA can get put into the cell, and then it uses the natural molecular biology of the cell to produce the spike protein. That's why spike protein can then be presented on the surface of cells for the immune system to recognize. So in terms of a safe vaccine, the trials, there were at least 3 levels of trials to show whether there were any serious side effects. Those came out quite significantly, showing that it was a safe vaccine. But importantly as well, that it was very effective. So I think the last trial had 38,000 people in a number of countries, and then an equal number got a dummy vaccine, and the number of cases in those that got the dummy vaccine was vastly more than those that got the real vaccine. So that's where the number of 95% protection comes from. Now, the important questions, however, are two. First one is, what happens to MECFS people if they actually get COVID-19? And there is quite a few people have actually got the infection, and there's one study that came out last year I've seen, which I'll speak to in a minute. And what happens afterwards? What happens to MECFS people after they've had the infection and they're in the sort of recovery period? And I'll speak to that. The second question is, what about the effects of the Pfizer vaccine itself on MECFS people? And I'll talk about some of the numbers that have come out from patient surveys. And so then just finish off by saying, well, how can one make a decision then to actually whether to get the vaccine or not? So in terms of how are MECFS people affected when they get the COVID-19, I was rather surprised that it's only one report that I've seen that's come out from the United Kingdom in 2020, and that was they weren't more seriously affected than the general population. That is to say, they weren't sub-selected into that group that have to be hospitalized or in ICU. So that's mildly good news, that it's not affecting them more seriously at the time of the initial infection than the general population. So, secondly, what about post-COVID? And there, the story isn't quite so optimistic. About 40% of people reported they were back to their pre-COVID health, that is to say their restricted health stated, which they had with MECFS before, but about the same number reported they were either moderately worse or much worse than before the infection. So the post-viral fatigue syndrome for about, should we say, half the people gets worse and half the people are lucky and it doesn't get worse. Now, in terms of taking the vaccine, there are two patient survey studies I've seen. One just came in to me in the last couple of days. They have slightly different numbers. So with the first dose, a significant minority, the first study came out at 30%, 10%, the second one is reporting 30%, experience a significant relapse. The second dose, about 30% also with severe side effects that are ongoing, lasting about 30 days. So kind of what these patient surveys are saying is that, almost splits into two groups. About half the people don't seem to have any serious side effects from the vaccine, and they get a response which is rather similar to the general population. But unfortunately, half get much more severe effects. And so this is a bit like tossing a coin, and it makes it very difficult if you're an MECFS sufferer, I know, to make a decision on this. So a lot of people, as I said, have asked me about this. And so what I do is to try to go through their history of whether they've had vaccinations during their MECFS period. And it seems to split into two. Some of them say, well, they may be a sort of feel off color for a couple of days, but normally okay. So that's quite a good response. Others say they've been knocked down for weeks or even months. And so that's not such a good experience. And so from that kind of history, I've been saying to people, well, maybe the risk for you is low, or maybe the risk for you is high, and maybe you should make your decision on those kinds of things. But of course, it's up to the person themselves to make the decision whether they're going to take the vaccine or not. One thing that has been suggested by clinicians, certainly in the United States, is that if you decide to get vaccinated, a good thing to do is to take a strong antihistamine, maybe a couple of weeks before you get the vaccination and a couple of weeks afterwards, so as to suppress mast cell activation, which can lead to relapse and severe fatigue type symptoms. I've not seen any reports of the results of that. They're just opinions from clinicians that this would be a good thing. So I just put that forward as a possibility. And we might say that we think antioxidants, taking an antioxidants like coenzyme Q10 or mitoQ10, N-acetylcysteine, if you can get it, these could be somewhat protective as well. Okay, so now I want to just switch tack and talk a little bit about, my history with this disease. And this started over 30 years ago when my younger daughter, age 14, after an attack of glandular fever, a month or two later, came down with a severe fatigue illness with lots of different symptoms, which we now know was MECFS. And interestingly enough, the clinicians at the time in Dunedin really didn't have any idea what was going on. And that's despite the fact that a few years earlier, we had a mysterious outbreak in a South Otago town, which you may have heard of, called Tapanui, which has subsequently been called Tapanui flu, which led to ongoing disease. And what I discovered as a parent was that if clinicians didn't have an idea when they went through their sort of formal diagnostic protocols, then it tended to go to a default view, which that this must be a psychological psychiatric disorder. As a biochemist, what I was particularly struck with was this pervasive fatigue, and I thought there had to be something wrong with the energy production in the body. So we had an expert on energy production in mammalian cells in my department at the time. So we put in a grant to the Health Research Council, and it came back with the reviewers saying, this is not a real disease and you won't find anything interesting by doing this. So the grant was turned down. In fact, that was then 20 years before I was able to get started to work on research on MECFS. Now, a person that had been a dominant figure during this sort of period from about 1985 to 2005 in the United Kingdom, it spread to Europe and also, affected, I think, attitudes in New Zealand, was this psychiatrist, Sir Simon Wesley, a very distinguished psychiatrist, very influential in the UK health system. He was president of the Royal Psychiatric Society, and he thought that the work he had done on ME was the best work that he had done in his career. And his conclusion was that ME is a perception disorder. That is, that patients perceive their symptoms and therefore they perceive they've got disabilities. And when they look at things they have to do in life, it all is too difficult. So it becomes an opting out syndrome. Not surprisingly, UK families who were ME families became quite angry at this because it essentially dismissed what they saw was a real illness. And surprisingly, he accepted the fact that the origin was often sort of a virus. In other words, there was an organic cause at the beginning. And in the end, he started to get death threats. So he stopped working on ME in about 2005, but his legacy has gone on really even till now. And it followed with a study in the United Kingdom called the PACE study, which was published in the Lancet Psychiatry, which concluded that the best therapeutic options for ME patients to recover from their disease was graded exercise and cognitive behavioral therapy. And that has been on the recommendations all around the world ever since until this year. And in fact, just this year, the National Institutes for Health and Care Excellence in the UK, NICE, has revised their guidelines, and so now graded exercise therapy and cognitive behavioral therapy have been withdrawn as treatments of choice for MECFS patients, because they said they didn't find supporting evidence when they examined everything that exercise and talking therapies would increase activity levels for MECFS patients. And of course, they found that for some patients, it was an unacceptable risk, because we know with post-exertional malaise, exercise can, exacerbate the illness and cause serious relapses. And also in the United States, the Mayo Clinic has just published a paper in the last couple of months with 21 outstanding ME clinicians also actually saying that these things are now not being recommended as therapies for MECFS. The residual sort of tail of this in New Zealand is seen because a number of patients at the moment that I'm getting contacted from are getting insurance is refused because when the insurance company consults with, in this case, I think it's mainly a psychiatrist, they get told this is not a real illness, and so therefore the insurance company doesn't have to pay out. So we've still got a little bit of work to do, but 2021, I think, has been a watershed year in casting off this kind of blanket, which has really been over MECFS. I just wanted to say something about why we should have perhaps had a much better idea of MECFS earlier, because since 1930, there have been 75 outbreaks of infectious diseases, probably viral illnesses of unknown origin, that have caused ongoing post-viral fatigue syndrome. So it's not a new phenomenon. In my lifetime, year I was born in 1946, in Iceland, there was some cases, Northern Iceland. And what I want you to notice is the number of cases here, about 1,000 cases. It was called the name, I won't try and pronounce that, but that's the name of the Northern Iceland town, and it lasted about 3 years. And interestingly, in 1986, 40 years afterwards, people went back to try and find some of these people and see whether they were still had the illness or had recovered. And about 80% of those that they found still had ongoing illness. Perhaps the one that's received the most publicity was at the Royal Free Hospital in London in 1955, where you can see there was just almost 300 cases. And from that's where the name ME came from. So obviously the symptom, myalgic and cephalo and the itis, the inflammation, must have been appreciated at that time. In Incline Village, Nevada, on Lake Tahoe in 1984, about the same time as we had Tapanui flu in New Zealand, that was a village of about 900 people. They had 175 cases. So the two GPs in the area had queues of patients with severe fatigue waiting outside their doors. And they called in the Centre for Disease Control to come over and actually look at this. And the conclusion in the report that was made, this was a case of mass hysteria. But I think this is what gave rise to the name of chronic fatigue syndrome. And that's why the disease was called chronic fatigue syndrome in the US for a long time. And of course, now we use the name ME/CFS, and that's generally accepted quite late in the day, only really about 5 years ago, where it was internationally accepted this was a good way to refer to the disease. But what I want you to do is to contrast that with the current pandemic. We now have about 240 million cases, you know, what I'm sure of the actual infection, and a conservative estimate about 20 million cases of the post-COVID-19 condition or long COVID. So 20 million versus just the hundreds of cases, or just over 1,000 we saw in the first one there. And I think this is why suddenly everybody's been shaken up by this and become so much aware, because there's so many and it's all occurring at the same time, whereas in MECFS, of course, the infections have occurred from time to time and they've been relatively small and geographically localized. So in 2012, then I got back to starting to work in MECFS research. And as a biomedical researcher, it would be normal for me to look at some aspect and work on that with great intensity to try and understand some feature of the disease. And that's what I've done with Alzheimer's disease. I'm working on a protein which is neuroprotective in the human brain, and we think it's got a lot of promise as a therapy for Alzheimer's disease. So that's a highly focused, classical way as a biomedical scientist you would function. However, you know, and also influenced by the fact that I was part of an ME family, it struck me with the myriad of symptoms in this complex disease of MECFS, and with the big questions unanswered. You know, why do a certain percentage of people that get a virus illness come down with the disease? Why don't they get better? Why do they suffer from relapses? These are big questions. I thought a different approach was needed if one could try and get an integrated picture of the illness. So I looked at all of the individual pieces of research, and they were like pieces of a jigsaw, but they didn't tell me what the illness was really about. And so it struck me that I needed an approach where I could actually get an integrated picture. And so I was inspired by a geneticist at Stanford University in the United States, who did a very self-indulgent study on himself. He had blood taken from him over a 2-year period, and he looked at all of the molecular classes of molecules in the blood sample over each sample of the blood, what we call multi-omics kind of study. Now, this was a multi-million study, and there were 40 authors on the paper, but it struck me this is just the approach we needed to actually try and understand MECFS. The problem is I didn't have multi-million dollars to do the research, and I didn't have a research team that was kind of made up of our 5 million, as it were, in New Zealand. So we had to think of a way how we could mount this approach, but you know, in a much smaller way. And I looked at actually the study of rare diseases as a model, where there's only a few patients often. And I've got an ex-student working with a company looking at rare diseases, and he was very encouraging. He said, if you study a small number of patients very intensely and do appropriate statistics, you can learn a lot about them. So this is in contrast to taking lots and lots of patients, thousands of patients, and trying to look at characteristics and then see what's characteristic in the disease. It's called, used to be called a personalized medicine approach, now called precision medicine. And so I started my studies then doing 10 patients and 10 age gender matched healthy controls. And that's what I'll just give you some little vignettes of the research findings that we found with this. And I wanted to say first, to be able to do this successfully, the patients have to be diagnosed very accurately. And this is a problem with MECFS patients all around the world, because there are 20 different clinical case definitions and people are using different ones in their studies. So it's very hard to actually compare studies. But we're just so fortunate in New Zealand to have Ros Bellings as an expert GP and she's diagnosed all the patients that I've used in my studies. Interestingly enough, she first encountered the disease in 1955 in the Royal Free Hospital in London when she was in medical training, and that alerted her when she started getting patients coming into her practice, her GP practice when she was in New Zealand, that it might be the same disease. So I can't speak more highly of her, and we communicate daily really, and she's been just a huge strength for me in this research. So we had a limited amount of money, and so we had to take a very simple strategy, and the strategy was to take 20 mils of blood from each patient, and we isolated the immune cell fraction, the lymphocytes and monocytes. That's the white band and the tube in the middle of that picture. And we isolated the plasma. And so from each of these patients, we did multi-molecular studies, multi-classes of molecules, just like the Stanford example. And my technician of 40 years, Tina Edgar here, she's fractionated lots of blood samples now from lots of MECFS patients. So I just want to talk to you about some of the studies we've done on these immune cells here and give you an idea exactly what we've managed to find out. So the first study, we looked at the expression of all of the protein encoding genes, particularly, which essentially the proteins are the worker molecules of our cells. They do all sorts of tasks. They're like the workers' bees in a beehive, really. And so Erin Sweetman here did her PhD on this, and she analyzed 13,000 transcripts. Now we have about 20,000 protein encoding genes, so this was a lot of transcript, and she got huge amounts of data. And I'm just showing you here the conclusions that we came up from by analyzing all of this data. What we're able to show was that there was defect in the expression of genes involved in energy production. And that was back to my original thought 20 years before, that there had to be something wrong with energy production. We also showed evidence that of oxidative stress occurring in MECF patients. There had been a Stanford study in 2016 of metabolites in plasma, and they indirectly concluded that there would be a decreased metabolism in about 20 biochemical pathways. And we saw in this complementary study by a different technology, a lowered rate of metabolism. And the Stanford people said it's like ME people are in a state of hibernation. And we now think of it, it's like their bodies sense physiological danger and they go into a kind of a shutdown because that danger signal is still being detected. And my big overview thought at the moment is if we can learn how to switch that signal off, that we might be able to actually reverse the whole disease. The other thing we were expecting to see was evidence of immune dysfunction, and certainly we saw lots of that, and also disturbed inflammatory pathways. The one that was a bit of a surprise to me, and perhaps it shouldn't have been, that there was defects in circadian clock function, which controls circadian rhythm. And well, non-refreshing sleep is of course a common problem with MECFS, and this is part of your circadian rhythm function. And so this certainly relates to that. Well, Erin stayed on with me, and we wanted to now look at the actual immune cell proteins themselves, what's called the proteome. And by the most sophisticated technology, just to look to see the amounts of each of the proteins that are normally produced in very carefully balanced ratios, so we have a perfectly functioning cell. We wanted to see whether, in fact, it was disturbed. And so Erin isolated all the proteins from the immune cells, and she digested them up into tiny pieces. And then we did this was a 10 patient and 10 healthy control sample again. She took a small sample from each of the patients and controls to make a library of all of the proteins that we could detect, and that was about 3,000 proteins. So she was looking at that. And then she did a sophisticated mass spectrometry analysis, so we could look to see in each patient how each protein was being synthesized and whether it was in normal or abnormal amounts. Now, this was a huge amount of data, but one way of stratifying the data is to do what's called a principal component analysis. So this is a little bit science geek rich, but I think it's worth sort of you just getting that sort of overview of this. So what we were asking here, if we do this analysis, can we separate out the individuals into groups? And what you can see is some red dots, and that's all of the patients. And you can see some blue dots, and that's all of the controls. And so we were really excited about this because it said we could separate out the patient groups and the control groups on a control group on the basis of how the proteins were being synthesized. And it turns out we got a medical biostatistician to help us to see how many we could separate, and 9 of the 11 patients separated out from the controls. So what were the proteins that were different then in the ME? And it turned out in healthy controls, there were about 100, in fact 99 to be exact. And half of them actually came from the factory in the cell where energy is produced. So this gets us back to energy production again. And so what you see here, again, it's a sort of a slightly complex looking diagram, but this is a cartoon of the factory in the cell called the mitochondria, where energy is made. And so we eat food and we break down the food by metabolism to produce high energy molecules. And this is produced here in the final step. And that goes into the first workstation of the factory. And then it gets processed along here until at the end, we make the energy molecule, and that's actually called ATP. So this is basically, we make a huge amount of this every day, and that's what allows us to keep going. And so what we're saying with MECFS patients, we wondered whether there was a deficiency in how this was made. Now, all I've told you so far is that these factories are made up of proteins, because the proteins are the workers of the cell. So that the amounts, the carefully balanced amounts of the proteins in these complexes and the structural things of the protein and of the mitochondria and of the factory and of the metabolism, they're all in the wrong amounts. And so what we wanted to know, the next thing was, does this affect the function of the synthesis at each stage of the process before we get down to this energy molecule? And the way we do this now, because there's fortunately a new piece of equipment available called a Seahorse Analyzer, where we can take live cells from patients and we can measure each of what's going on in each of those steps here of the pathway to making the energy molecule. And in fact, these colors you see here are the things that we measured. I'll show you a little bit of data in the next slide, but it's basically measuring various functions of the energy production to see if we can see if any of these are not working properly. Okay, and so we'll move on here. So those colors then, those colored blocks convert into, once we've got the analysis, into small graphs. And I've pointed out 3 here that have red rings around. So here's the MECFS group, and here's the control. And you can see here there's a trend for the energy molecule production, which is lower in MECFS than control. Not too much, but it's lower. But we're not actually putting these cells under stress too much stress when we measure it. There are 2 other things here which were quite indicative, and that is that the level of basal respiration, how much oxygen was being consumed by this factory in the MECFS group controls was much higher. And this says that somehow that factory knows it's not producing enough ATP or producing it properly, so it's trying harder to do it. It's consuming more oxygen. The other important factor was that this thing called spare respiratory capacity. And as we go through our daily lives, if we actually come upon some stressful event that actually stresses us, then we actually need more energy to cope with that stress. And so we need spare respiratory capacity to deal with it. And so if we have less spare respiratory capacity, then there's a chance that we won't deal with the stress so well. So that's, of course, a characteristic of MECFS, is that very sensitive to stresses. So this was quite an exciting result. It's not, we get a span of data from individual patients, individual controls, but the trends are suggesting that ME people do have dysfunctional mitochondria. They're not quite as good at producing energy. They're working harder, and they're not so good at actually dealing with stress. Now, all of this then has told us that there's a lot of dysfunctional physiology, but at what level is all of this controlled? And we know now, just apart from the genetic code, which I'm sure you've all heard of, that's the G, C, A, and T bases of the code, which is on, crime series when you do DNA analysis of a crime scene. That's the genetic code, and that doesn't change during our lifetime. Each of us has a unique genetic code, very close to our siblings and our parents, but a little bit more different from our friends and neighbors. But we have another code, which is called the epigenetic code. And this is like a series of light switches or dimmer switches, millions of them spread throughout our DNA genomes. And those are all set, and they can change during things happening in our life, but it's set to control in a very balanced way all of the proteins that we make for the myriad of functions we have to carry out in each of our cells. And so we wondered if that was the source of why MECFS people were making different amounts of proteins and therefore having different functions of some of the important things that are going on. So I recruited 2 young university women with MECFS in my university woman study group. And I asked them whether they'd be prepared to have blood taken over a year, and but they had to promise me one thing, and that was they had a relapse during that time. And I was amazed that actually 2 people agreed to do this. So I can show you this here. So here's patient 1. And so I'm just showing here 5 blood samples that we've analysed in depth. So the first sample, this patient, I asked them to say how they were feeling. And so in this case, this person said they were feeling well for the first sample and ranked themselves 7 on a scale of 1 to 10. By the next blood sample, she said she was fragile. And although I told her only from between 1 and 10, she ranked herself -3. So that was a pretty severe relapse. She still had the relapse in the next sample, and then her health returned to near before relapse levels, although she ranked herself satisfactory, gave herself a 7 in those 2. The second patient had more fragile health right through, ranking herself 5 and 6, did suffer a relapse also, down to 2, and then recovered and was going into another relapse here with the last blood sample. And then our healthy control was obscenely healthy, well all the way through, and ranked herself 10 out of 10 all the way through. So we were really looking now, this is what you call precision personalized medicine at an individual patient and comparing them across the longitudinal study, this one across the longitudinal. We're also looking at the 2 patients versus the control. And we discovered one thing which was quite a surprise, and that is this epigenetic code, this careful setting of the light switches and the dimmer switches was much more unstable in the 2 patients than it was in the control. So when we went from month to month, even when they were relatively healthy or relatively well for their illness, there were about 2 or 3 sites every 100 over the millions across their DNA that were changing in both of the patients, whereas the control was very stable, only about 1 in 1,000. So that was an interesting finding and suggested, that the instability perhaps of health that MECFS people are having is partly because their epigenetic code settings are changing much more rapidly than the healthy controls. Now, what I'm showing, so this is the solidest data I'm going to show you, but this is now where we're looking at individual sites on the DNA. So each of these little boxes are one site on the day and on the DNA, and I've highlighted 6 for each patient. And the green one is patient 1, and the shading is when she had her relapse. And what you can see is the setting of the dimmer switch here. It's almost completely on. It actually gets reset during the relapse down, and then it recovers, resets down and recovers, resets down and recovers, and the same with the other 3. With the other patient, who only had a 1-month relapse, we see it's a lot sharper, therefore, sharp down and then recover, sharp down and recover. And she had some that went the other way, up and then down, up and then down. So these are individual sites of light switches or dimmer switches on the DNA which are changing during the relapse and then recovering. And there's a lot of complex data gone into this complex diagram, but the only thing that I want you to kind of realize is that actually when the changes in the 2 patients were different. That was interesting. They didn't both have the same changes, but when we analyzed the consequences of it, they had the same effects on their physiology. In other words, mitochondria, which means energy production, this came out with this. So energy production is worse during a relapse for both patients. Metabolism, effect on just this going into hibernation, if you like, effect was worse for both patients, even though it was from different causes. And then we could show immune deficiencies as well. So we did another study, which was now a 10-patient, 10 control study. And I just want to show one piece of information on this. We found 5 genes. These are them here. I won't go what they are, but they affect the release of neurotransmitters in the brain that impinge on the hypothalamus. Now, some of you may have heard about the HPA axis, the hypothalamus pituitary adrenal axis, which is known to be disturbed in MECFS. So we found a molecular signature around 5 genes which affects those transmitters being released onto the hypothalamus, and that's where the stress center in the brain is as well. And that will affect the response of hormonal responses and the production of cortisol. Now, cortisol is known to be low in MECFS, and that's associated with fatigue. And there's a study which suggests it's associated with post-exertional malaise. So what these molecular studies have told us is that we've found molecular signatures in proteins that are responsible for energy production. So that's a molecular signature for MECFS. And we found molecular signatures in the DNA for proper neurotransmitter release and therefore control of the hypothalamus pituitary adrenal axis, which deals with the stress response. So we're starting to get there now, a real understanding of why the disturbed physiology occurs, what's really at the molecular level causing the effects that many of you are experiencing. Now, I just wanted to talk about what we're doing, just overview in 2021 and 2022. We're now involved in the most extensive study yet of post-exertional malaise that I think has been done anywhere in the world. That, of course, is a core symptom of MECFS, where people, if they have physical or emotional or psychological exertion, then that can often lead to a relapse in health, post-exertional malaise. We started this with Lynette Hodges, who I gather talked to you last year, came down, and in our selected patients, she did cardiac physiology with them, and we took blood during that, before and during and after exercise. And then we've looked at what happens to a lot of their molecular biology as a result of that. And unfortunately, with the lockdown, and we're about 3 or 4 months behind that now, so I don't really have definitive data to tell you, but we have seen some effects. I'm also involved in a research project on long COVID, where we're doing a comparative study of MECFS patients and long COVID to see whether they have the same molecular signatures, because in my view, it's very likely that these are one and the same disease, except long COVID actually comes from a specific virus, namely the SARS-2 virus, whereas MECFS, of course, comes from a number of different viral infections, but also can arise from surgery or childhood trauma or agricultural chemicals. So it has a kind of a broader initiating stressor, but the physiological response, and certainly the clinical case definition just released last week by the World Health Organization, almost exactly mimics the clinical case definition, which is accepted now for MECFS. So this, in my view, is going to likely to be seen to be, if worst, a specific class of what we classify as MECFS. So we're looking at a lot of molecular parameters in that. And then I'm excited to be collaborating with Dr. Anna Brooks, who's an immunologist from the University of Auckland. We've been talking for the last 6 months and planning studies, and we're interested, of course, in one of the things we're interested in, the effects before and after vaccination of people that have chosen to be vaccinated, to see if we can see changes. And I'm excited to actually get immunological data to back up the molecular data that we can gain. So this, I hope, will be a very fruitful collaboration for our work to go on in the future. And thanks very much for your attention. And I acknowledge all of these people that have been helpful, and particularly the financial support from ME families and private donations. Without that, we wouldn't have been able to do these studies. So thanks very much. And I'll stop sharing now.
Oh, wow, that was absolutely fantastic, Professor Tate. You know, I've heard about your research and I've read a little bit about it, but to hear more detail from you is absolutely fantastic. And the information on the molecular studies of your immune cells, the proteins in those cells, and the genes that affect the neurotransmitters in the brain, it all, you know, having my ME myself, it all makes sense. And it's really, really exciting to be able to, you know, see some graphs that concretely show people how things are changing within our bodies that are different to what we say normal, healthy, controlled people. Really looking forward to the results of your post-exercise malaise studies and the long COVID ones, and I really hope that you're able to kind of push on with those soon. I know it's been a massive interruption for you and for your students. I'd love to invite some people to ask some questions now, if that's okay with you.
Sure.
Libby.
Oh, hi, everybody. Thank you, Warren and everyone. Yeah, I know a bit about the future studies with Anna, and Anna and I are in close contact. And I've been on the ANSME's executive committee
Yep.
since 2005, but it was like, anyway, there's a problem that I don't know if you can help us with, Warren, and this is basically when you spoke about, and this has to do with advocacy. You spoke about the fact that, of course, one could say that people with ME have every right to say, I can't be vaccinated, it's too dangerous for me. The problem I think we're having is there's a gap between IMAC, who's the Immunization Council, there's a gap between clinical practice. So until IMAC, there are people that we represent who are working and who are needing to deal with exemptions. They're needing to deal with people like me who can't actually isolate safely. So what I'm wondering is there a possibility that you could somehow help us with IMAC and explain the situation? Only because I've been working on this for 6 months, and there's a patient in Norway named Monica Vorland. And I think I've given all this to Roz and Anna. Monica chose, because of her situation, she needs a vaccine passport, her family and all that. She chose a fractionated dose of the vaccination attenuated schedule. But Monica presented her results were just simply the antibody test, right? The one we can all get easily. And she was turned down by, IMAC is called CISVOC over there. CISVOC said, oh, sorry, no. So in other words, until IMAC can show us what we need to prove for an exemption, how can such an exemption be given? And how can an alternative be accepted for those who do choose a fractionated dose of the vaccine? In other words, I'm hoping to present, I was doing it on my own with Pfizer Medical Affairs, hopefully, but then I found Anna. So in other words, how can someone prove to IMAC that they have achieved the same thing as 2 doses of the vaccine? That's where I think we're having a problem. And I don't know if you can help us with that or not.
Well, I'm certainly, in fact, it's interesting that I got contacted during the week by someone at Otago who's an immunologist, and we've started a dialogue, and I've kind of, we see things differently from our perspectives, of course, and so I think there needs to be dialogue. I'd be very happy to, for example, make contact and offer to be in dialogue to just see if we can get a better understanding and appreciation. So a couple of things, what you said. I mean, I've been very disturbed because now there's this hype, and this weekend, of course, to get everybody. I'm all supportive of that, but people to say very loose things like there's only 100 people in New Zealand that would need to be exempt from getting vaccinated. Well, that's sort of crazy.
Right. It's not right.
And so what I think is needed, however, I mean, what I debated with this person this week was, would a 3rd dose be good for MECFS patients? And he said, well, as an immunologist, that would be good for me because, that's what's going to be used in children. And I said, as a biochemist, I don't, we know, let's say up to a half patients can get quite severe effects from the vaccine. I can't tell you that a 3rd dose, would stop those side effects. There needs to be a proper study done,
There does.
Sort of dose where you're looking at efficacy versus, seeing what the side effects, and then one might be able to come to a very sensible conclusion that, a dose could be good for MECF patients.
I agree. It's got to be, they've got to have some kind of proof. It's got to start somewhere. I've been, working on my own, trying to write an amendment to the study and all those things maybe Anna has told you. And I don't know if that's going to be possible. But, you know, my situation is such that I'm willing to go ahead with some kind of fractionated dose. And you can get it through, they finally set it up through Whakarongo. So you can have clinical care, because I don't know, you know, Nancy Klimas, you may know who Nancy is.
I know.
She said, okay. So I met her too. She said, okay, well, in this case, a physician may give a patient a steroid in advance to manage the inflammatory response. Well, of course, my GP is like, oh my God, that's horrible. What a terrible thing. I'm like, well, please get educated. I mean, it's exhausting trying to educate people, you know, to have a GP who says, well, I couldn't do a thing like that. And I'm like, well, why not? You know, would you rather me be really sick and have to go into, you know, nursing care, or would you rather help me? So it's quite challenging, but I think the only way through, and when I finally spoke with Pfizer, Pfizer's like, okay, send us a study, right? Because I've read the entire clinical trial. It was done on healthy people, and we all know that. It's quite specific. It's quite specific who is included, who is excluded. I don't see how anyone could argue that we don't need an observational study. It's just a matter of getting it done and getting people who are willing. For me, it would be a fractionated dose of my choice with a period of time to observe if I relapse or don't, because I'm not willing to trade the current function I have for that.
And sorry, can I just briefly?
Oh, I'm sorry.
No, that's okay. Just to add to what you've just said right now, that someone who contacted me said that if I took the vaccine, you know, as my family want me to do, and I got worse, I couldn't live with that.
That's right.
You know, that was
that's right.
And I understand that. So I'm just affirming what you were saying.
Yeah, thank you, Warren. Yeah, I'm sorry. I've been resisting writing emails to you and Roz both, because I thought I can't do this to them. And, you know, because I just didn't want to add to anyone's burden. But again, it's true. I do forget that healthy people can take a lot more than I can. So,
well, can I just that? I mean, Roz and I have joked and talked about this, and says we both have to live to at least 130 because it's to do. And I have to say, I didn't expect, you know, at my age, you know, I'm retired, of course, but I'm working full time, and I'm very excited to be doing it because it's such a good cause. I hope that, you know, each day I get up in the morning, I hope I've got enough energy myself to carry on.
I feel the same. Thank you. And well, what I'll do then, I don't want to take up everybody's time, although I feel it is important, and there's so many people who could use an alternative to, and also people will need exemptions. And I think if we are more proactive, and for example, Monica Vorlin in Norway is going, now what? And I said, well, just don't give up, Monica. You just go in there with your doctor and insist that they have to tell you what you have to prove. The burden of proof is not on you. The burden of proof is on someone who's saying, what does this immune signature look like? What does it look like biochemically? So that's my idea. Let's see.
Thanks very much.
Okay, that's about it.
I'm just going to move on because we have got quite a few questions in the chat, and so we can take that conversation offline to deal with that stuff a little bit further on. Professor Tate, there has been a question here, and I know you and I have spoken about this. Someone sort of said if they react badly to the flu vaccine, are you indicating that the COVID vaccine could be something that they would also react to?
I would, you know, so my response would be that this might indicate a risk factor for getting a severe thing. So, you know, I would never say don't get vaccinated, but I'm saying you have a higher risk. And can I tell you one example? I have 3 of my young women in my study group who wanted to go overseas, so they've taken the risk to get vaccinated. One had no symptoms, you know, above what I had from the vaccine. One was down for about 2 weeks, but has recovered from her to a vaccination health. The other one was severely affected and was hospitalized. And when, you know, I investigated with her family the history, she has always had some problems with vaccines. You know, she's always had some kind of negative effect. So there does seem to be a correlation between just that ongoing negative effect and the negative effect that occurs with this vaccine. So that's kind of, it's a rule of thumb, but that's where I'm coming from in my advice. Whereas if someone said, look, yes, I have flu vaccine every year and it never affects me, then I say, well, maybe you're going to be in that 50% that don't have an effect. It's not a very, you know, precise.
It's at least give something for people to think about. Somebody has asked also what a strong antihistamine might be, you know, to ask the GP for. I have heard people talk about just getting Zytec from their local pharmacy, but did you have any other thoughts on that?
I read this was what Nancy Klimas had actually named a couple, which I'm not sure if they're same names in New Zealand, and I meant to actually have that with me, and I'm sorry I didn't, but I can look up that and actually send it to you if you like. But
yep, I can post it on our Facebook group and stuff.
I mean, Nancy is a very good sort of hand wavy kind of, you know, global kind of advice person, and I'm a little bit nervous sometimes with, you know, these global suggestions, because, you know, I want to see kind of, okay, is this going to really, is a strong antihistamine going to be all right for MECFS people as well, you know, because, you know, the effective drugs and things like that, there's that super sensitivity.
Someone else has also asked, given the research that you've done that looks at, you know, the changes or the mitochondria not sort of functioning well, I don't know if you have an answer to this, but someone has said, is there something that they can take that is going to support the mitochondria or support energy making in the cell?
I mean, one of the things I didn't say is that there's a whole machinery in that factory which deals with reactive oxygen species. And if they're not dealt with well, that's when we get oxidative stress. And oxidative stress is associated with disease, and we saw that. And so we have done some studies with MitoQ, for example, to see whether, and it did look as though it improved mitochondrial profiles a bit, but the individuals didn't feel any improved wellness from it. But it's kind of what I feel is actually taking an antioxidant like coenzyme Q, N-acetylcysteine, or MitoQ10. I think that's a good thing to do, but I would love to have more stronger evidence to support it, you know.
Sure, absolutely.
We did one of my university women took it for 5 months, and we could see a change between her before and 1 month, and then by 2 months there was an improved sort of function ongoing, you know. So we did see some molecular change.
Cool. So somebody asked, are the funded tests that we could order to check if we've been sort of exposed or have sufficient antibodies to COVID before the vaccine or after someone was going to do fractionated dosing or what have you?
Well, I mean, Anna Brooks, for example, you know, who's an expert immunologist, can actually check to see whether, could check, you know, has the expertise to check to see whether you've got antibodies or someone's got antibodies to the virus already. And I mean, in a way, you know, part of what we've talked about is that there are a number of people who now have long COVID, but never actually had a PCR test at the time that they had the illness. So the question is, did they get MECFS from another cause, or did they get long COVID from the illness? So, you know, immunologically, you can test whether in fact they're showing any antibodies which suggest that they were exposed to the virus.
But that doesn't give you a level of how much your protected against getting the virus again, does it?
No. It just is you've been exposed to it somehow, Yeah.
I've listened to things in the past that have talked about how mitochondria work, and it's suggested that we're either in an energy production mode or we're in a kind of a cell defense mode, kind of fighting things off. And is that how mitochondria actually work in terms of, and could it be that ME people are kind of in a sort of a fighting mode more than they're in an energy production?
Well, I mean, that's a good way of putting it. I think that's kind of what physiologically is happening, and mitochondria get damaged. And what we realize now, that mitochondria do more than just produce energy. They're like cell signaling sort of organelles for the body, and if they're damaged, they can leak out ATP, for example, and that can form a cell signaling molecule which signals danger. So they're playing a much more complex role than we thought. But I mean, what I was talking about was that our mitochondria seem to be working harder, trying to kind of, they're obviously getting signals they need to work harder, and yet they're not doing it as efficiently.
Yeah.
And I guess when healthy people are, have stressful events in their life, the mitochondria will be working harder to produce energy to deal with that kind of stress. So, but there's still a huge amount to learn in this area. So.
Oh, absolutely. Well, our human bodies are pretty intricate, aren't they? So, one other question I have here, and then I'll chuck it out to and see if anybody has any more, was if someone's not vaccinated, you spoke about family groups that the studies have done overseas, and say 4 in the family and the rest of them are vaccinated, then you're 90% protected. With COVID and it changing, and the fact that they're saying that even if you're vaccinated, you can still catch COVID, then does that still mean that somebody unvaccinated is protected in that family group, because the member of the family could still get COVID and pass it on to them even though they're vaccinated? Like, does that change it?
I understand totally. I think it's been a bit controversial, but vaccinated people who get COVID probably aren't transmitting as much virus. That's a critical thing. So, it's like initially when the pandemic came, you saw places like New York, were having a horrendous time. And I think it was because the air was so thick with virus that it was spreading all over the place. Whereas if you were in a place where there were fewer cases, then the concentration of virus around is less. So that's probably it's going to be the same in families. But, that's a level of subtlety which, you can't have black and white sort of answers to this. And I was just impressed by that Swedish study. I was amazed that, they were able to come up with those numbers. Now, probably people with MECFS or immunocompromised in Sweden they'll be looking after themselves. Naturally, they have to do that to have a certain amount of protection, but it's the person going in and out, into community, picking up the groceries or being in a workplace, and being exposed, that's then coming back into the family, So that's what one's hoping the protection will give to that person, bring it back. But your question is, we don't know really. We hope,
No, and because I think the majority of the ME community who are considering not getting vaccinated are wondering how they live a normal life, really, if they don't, which is the big question. And I don't know that there is a real answer for that.
No, and the dilemma is, do I take a risk of getting vaccinated and having worse CFS, or so that I can travel overseas or whatever? I mean, it's an impossible decision, isn't it? It's an impossible dilemma, And I just hope, well, we're getting better antivirals. For example, now the new drugs that have been announced, ME people could be, protected, if they're not vaccinated, could be protected with those antivirals, which seem to be pretty effective now, these new ones. So we may get better things if they can tolerate them. So, and the other thing, the first SARS virus lasted a year and then it just died out. Now, I was amazed. I went back and looked at the number of cases, and that there were only 9,000 cases in the world, even though there was a lot of publicity, So we've got this, 200 and almost 40 million now. The question is, will this virus just die down eventually? Will that be our savior? And I think people were hoping that it might have just had a rise and a fall, but it's of course, in its fourth iteration now. So
yeah. So someone else has asked around the spike proteins. So do mRNA vaccines provide instructions to cells to make their own proteins? And is this definitively switched off, or does the body become a spike protein factory?
A very good question. So what happens? The RNA has been modified so it will last a bit longer. RNA normally turns over very fast. It's quite an unstable molecule. So the RNA is likely to last. I don't think there's any absolute figures on this yet published, but it will last for hours or at the most a few days. So that it'll be a factory for those hours and few days producing the spike protein. And then the spike protein will be presented and the immune response will be mounted. Now, proteins, we turn over our proteins quite regularly too. I mean, quite a variety of half-lives. Some of the proteins can last for 120 days, but generally it's much shorter. So we're constantly synthesizing proteins and turning them over. So when we produce the spike protein, if there's any spike proteins just hanging around, the body will naturally deal with it and turn it over. So that's not a problem. And the fact is, the spike protein shouldn't harm us in any way. It's not the virus. It's just a part which the antibody can react to well.
Leanne Catchpole, I see you've got your hand up. Did you want to ask a question?
Yeah, well, make a comment or a suggestion, really. So I work for the Northern DHBs, and I've got a connection in with the vaccination program delivery, and I've been trying to raising the issue of people with CFS, MECFS, and seeing whether we can get a process to get the fractionated doses. But I was just thinking one thing that might help, because this is not something that that they've probably even thought of before I raised it, because they're not, people with MECFS aren't on their radar. I think they haven't thought about this group of people. I know they've thought about people with cancer and other immunosuppressant illnesses. We see that in the advice that goes out to doctors, that's got some, specific conditions that they mention and about how to manage them. But I've never seen MECFS mentioned in any of the guidance that's come out from the COVID, management team. So I've just got a suggestion that I think that we as a community could do more to raise their awareness of this group of people needing special consideration and perhaps for those of you that have had one dose of the vaccine and haven't reacted well, if you could ask your GP to ask the COVID management clinical director, for advice on how to, like whether you could get a fractionated dose or how to manage, or if you haven't had any dose yet, ask your GP to ask the COVID management team, for advice. And then this I think at least will get it on their help to get it on their radar that there is an issue that they need to address, and looking at what's required for this population, and perhaps even, because I know that they're not looking at very many exceptions. When I read the rules around exceptions the other day, it was they were saying almost nobody's going to get an exception, but neither have they actually looked at the needs of this population group. So I just think we, see if you can get and doctors listen to other doctors. So if you can get your GP to raise it as an issue with the management team, that just might help.
Who are the management team, Leanne?
Sorry, it's actually called the NRHCC. So
you told me that.
Northern Region Health Coordination Centre. So that's the one for Auckland. It'll be a different one in Waikato or a different one in Wellington. you know,
Whereas
so,
more-
but anyway, this is an Auckland meeting primarily, so I'm talking about, but if you live in the Waikato, then you could raise it with your Waikato's DHB's management team, you know. So there's a clinical director, and then they get their advice from IMEC, who we've talked about earlier, the Immunisation Advisory Centre, which is a part of Auckland University, which I'm noticing that Anna Brooks works in the same university, so that might be a useful link there. But anyway, I just thought, you know, perhaps through your GPs you can help to raise the profile of this of our needs, really, and that we need special consideration.
Yeah, I mean, I'll also have a talk with Ros Bellings about this, because, you know, I mean, she's got mana, at least as a among GPs, about this disease. So
That'd be fantastic. Thank you. Do you mind if we've got a few more questions coming in? Is that
yeah, good.
We've got one from David here. Do you think the epigenetic and biochemical changes you're showing at the primary level of disease, brackets virus hacking cell machinery, or downstream from, e.g. a secondary derangement of the immune system?
Well, yeah, this is a good question. In other words, I've been interested in the big question, and what's the primary thing that happens when a person kind of the body responds to initiate the ME response. And I think it's probably seated in the brain, and I haven't talked about our work with neuroinflammation, but we've done quite a lot of work with neuroinflammation. We are, you know, we're looking at really the secondary effects of what the body's doing, you know. In other words, when we see these light switches and dimmer switches changing, what is it that's making them change, you know? So there's a high level, and it's finding that sort of initial high level thing, and we don't really know that yet. Is that not a very satisfactory answer? But I feel, you know, it's like you've got an onion, and you're peeling off the onion skins, and we're getting closer and closer to the core, you know, and the epigenetic changes. You know, for me, are quite an amazing finding that we've taken off the skin, and we've seen that. But I'm not saying that's the actual key thing, which is switching people into an MECFS type illness.
Awesome, thank you. I think there's another question. Sorry, I just scrolled down more. Do you see a place for genomic testing of people with ME, and then up or down regulation of appropriate pathways using things like nutritional supplements or that sort of thing?
I think, you know, in principle, this could occur. I mean, one of the studies I'm starting with a master's student who's enrolled in a genetic counselling course at the University of Technology, Sydney, and I've been contracted to do the project with her. We're looking at genetic susceptibility factors. Why is it that, you know, 1 in 10 people that get glandular fever go on to get MECFS? Why is it that 10% of people who have infected with COVID-19, why do they end up with long COVID, even if they've had very, very mild things? There's no information about that at the moment. So if we can actually tease out susceptibility factors in people, we might be able to then, you know, be able to predict people who get an illness if they've got a set of sort of criteria in their personal health history, family health history, that means you could have immediate intervention. You know, and but you know, genetic testing would be, I mean, it's very sophisticated, the technology to do this. It costs, you know, quite a lot of money. It uses, and it's not applicable to sort of chem path labs. You know, what I've tried to do also with the diagnostic test is to find something we can use that would be in all chem path labs that a GP could prescribe and say, do that test, you know, and we've done some preliminary studies on one which seemed quite promising on a chronically activated immune system, you know, with a protein where it gets a phosphate group put on. So looking at a phosphorylated versus unphosphorylated protein, that's rather like an HDL, LDL kind of test that we get done now, the same principle, you know. So, you know, I think there is a need for sort of simple analyses which will help, you know, and then you can take sort of therapeutic things, not necessarily super complex, sophisticated technologies, which, you know, take a lot of time, and so the individual patient isn't really benefited immediately. We want immediate benefits.
Cool, thank you. Patrick, do you have a question? You've got your hand raised.
Yeah, thank you for that presentation, Professor Tate. Just a two-part question from my end. Part number one is that you had a slide on the impacts of COVID on ME and impacts of a vaccine on ME, and I think, sorry if I'm wrong, my recollection is that COVID is no different in terms of hospitalization for people with MEs and healthy controls, whereas the vaccine itself, you sort of see about a 30-40% relapse rate. Is that correct?
Yes.
Is there anything?
The COVID infection, we're just talking about the initial infection, how seriously ill ME patients were compared with the general population with the initial infection. And the one report I've seen said there was no difference.
No difference, okay.
The vaccine thing, of course, was post-vaccine. And of course, if you looked at post effects of getting the virus, that was quite severe in ME people as well. In other words, the ME got worse. So I'm not saying, you know, we can sit there and not worry about getting the virus because you're not going to be affected any more than anybody else, because it's a serious, you know, some people are hospitalized and some people are in ICU, and of course some people die, and so it is a serious thing. But I would have expected, given the immune dysfunction, that ME people would have been, you know, more selected to have to be hospitalized and go into ICU.
So I was trying to understand if the first part is, wouldn't a relevant comparator between the two be the rate of relapse for the vaccine versus COVID? So if you had COVID and 80% of people had a relapse with ME, and that was really prolonged, and then only 40% had a relapse with a vaccine, you know, I mean, I know you're choosing between worse and bad, but I mean, that would be the best way to make a decision, would it?
Oh yeah, of course.
Yeah.
I'm not. I wasn't saying. I certainly wasn't meaning to imply that it's okay to get the illness because it's no worse by getting the vaccine. Not at all. But I'm really just bringing facts, you know, that are available now, just so you can see the facts and what the issues are. But
go from perfect information. And just second part, I'll just be quick, is looking around the world, the strategy of elimination is becoming increasingly rare. It's a luxury that can only sort of be afforded by countries that can take control over their citizens, like China, for example, not in Western democracies. If I was to look at three comparable ones to us with about a 5 million population, you'd have Ireland, Denmark, and Singapore, and we're sort of pushing between 500 and 2,500 cases a day. Is that likely to be our future around those case numbers, and would that change the risk sort of equation for a vaccine?
You know, it horrifies me if it is, I have to say. You know, I mean, it's interesting in Denmark where they accepting, you know, a few deaths a week and you know, maybe 100 cases a day or something, and they're getting back and doing things. Now, you know, I mean, that's, as a member of the 5 million of New Zealand, that doesn't feel good to me that we, you know, that we just would accept that. So I'm hoping that, and yet I realize the dilemma of having everybody shut down and whatever. I'm hoping that we can do better in a way.
The future's uncertain. I just ask because I work for government and we put together all the big forecasts for economy and things like that. And our working assumption is having, you know, a live off a virus strategy, and it's based on chats with a lot of stakeholders. I've noticed a huge tidal shift in my work over 3 weeks about, you know, just when is the government going to open up, and, you know, how are we going to live this thing? And my view, without knowing exactly what's on Jacinda and Ashley's hearts, is that they are going to be opening up next year, and it's going to be, we're going to be in a similar situation to other countries.
Yeah, I mean, certainly all the modelling would suggest that is the case, and particularly if we still have pockets of the New Zealand population which have lower rates of vaccination. I mean, it's interesting how we were saying at one stage if we got 70% of the population vaccinated, we'd be in a good state, and then it was 80% and now 90 and 95, because the modelers keep telling us, the consequences still of having 90% vaccinated is a lot of cases, and with the consequences. And I guess the only thing is that if those cases would include some of the people that are vaccinated, maybe a lot of them will be very low level symptoms. I guess that's the, and so the 100 cases we get might be mainly asymptomatic, of course.
Oh, thank you very much, Professor Tate.
Fantastic, thank you. There is one other question here. Have you tested for lactate dehydrogenase or found any slightly low alanine in patients at all?
Wow, that's I haven't done that. I mean, initially when my daughter got ill, we had an amino acid. I did an amino acid analyzer in my analysis of my work for things like that. I mean, lactate is produced more readily in MECFS patients, so and I didn't say that in some of the data there. You could see that non-oxidative methods for producing energy are used earlier. And like when you sprint, we use what's called anaerobic metabolism, and in a sense, MECFS patients are going around sprinting when they're just going around doing their normal lives, because they are using some anaerobic metabolism and producing lactate. So yeah.
Interesting, fantastic. Is there any last questions for Professor Tate before we wind up? Wonderful. Well, thank you so much for taking the time out of your Saturday afternoon. I personally feel it's been extremely worthwhile to be inside and listening to you. I really, really appreciate the time, and I'm sure everybody else has appreciated the time that you've given us today. So thank you so much. And we've got a small gift for you, which we will put in the post for you. We can't hand it over Zoom. But,
thank you.
I hope you enjoy the rest of your weekend, and everybody else, thank you so much for coming. Enjoy your weekend and stay safe.
Can I just say one thing? I've really appreciated the deep and thoughtful questions that came. I mean, it really, it helps me enormously to have this kind of conversation with people who have not only got connections, but also are living with the illness, because it just uplifts and sharpens my ability to interact with other people on it. So I really appreciate that.
Awesome. Thank you so much. Well, Saul, do you want to say something?
No, I just wanted to say thank you very much there to Warren and to everyone that participated in today. I think it was very worthwhile, and thank you, Kate, for putting all the work in, really, and Patrick. So yeah, thank you.
You're welcome. Thanks, everybody.
Thank you, Kate.
Take care.
Bye.
Bye, everybody.